Scientists are interested in how viruses figure out how to infect new hosts because that's the process that underlies the emergence of many of the scary pathogenic human viruses. You've heard of swine flu and bird flu, right? These viruses figured out how to jump to a new host, us, from their original hosts. The process that we saw in Lambda, recognizing a new host protein, is the same kind of process that underlies host switches in zoonotic viruses. So the next step in our research is to figure out whether this buy one get one non-genetic variation in lambda phage, our laboratory model, is common in other bacteria phage. That's where you come in. We want you to find out if other phage species produce non-genetic variation. How can you test if a particular phage produces non-genetic variation? Remember how our phage produces two different types of progeny? One type that can bind only the old receptor and one type that can also bind the new? Well, lucky for us, these two types are also different in another easy-to-measure way. It turns out that they have different stabilities. Stability refers to how long the phage can stay viable or infectious after it's produced. We found that the phage variants that can bind the new receptor are less stable than the variants that can only bind LAMB. We think that the receptor binding protein, which acts like a cork to keep the DNA inside the phage particle, kind of sitting at the end of the phage's tail, we think that that cork, this receptor binding protein, is more likely to denature or unfold and fall apart in the phage with the new function. If this happens, the DNA escapes and the particle can no longer affect hosts. On average, each phage type has a characteristic rate at which they decay. That cork protein denatures and the phage becomes unviable. After an hour at a given temperature, something like maybe 40% of the defendable LAMB binding phage type might decay, but over that same time window, you might lose 90% of the novel AMPF binding phage type. You can easily measure how many viable phage you have before, during, and after incubation by taking samples and plating them to determine your PFUs, plaque-forming units, per mil. But unfortunately, you can't easily separate the two types of phage. Remember, when they're produced during an infection, both types are made, so they're mixed together in the lysate. Fortunately, you can still see whether there are two different types of phage present by looking at the particular pattern of decay. When the stable and unstable types are mixed together, all of the phage will start dying, but the unstable ones will decay at a faster rate. As the experiment proceeds and the mixture runs out of unstable phage because most of them have decayed, your measurements will start to be dominated by the remaining, more stable phage type, and you'll see a slower rate. So this pattern where you have two phases to the phage decay, a fast decay phase and a slow decay phase, is called bistability, and that's what you'll be looking for with your phage. By testing whether or not your phage is bistable and sharing the results, we'll get an idea of how common bistability is in the phage world. Eventually, we want to see if this bistability allows phage to infect more types of hosts. So you might also be able to measure your phage's host range on a panel of different bacteria. By testing a lot of different phage, together we'll be able to get an estimate of how common this mechanism of evolutionary innovation might be.